Today, I want to talk about some invertebrate pain models and some cases of pain dissociation that can inform our definitions of pain and what is pain. So the first study we're going to talk about is a study conducted in Mexico in the early 80s, conducted by Maldonado. And what we are looking at here is a mantis shrimp that is a couple inches long, and it's tied up in a contraption, and they exhibit this tail flick response that is entirely electrical and automatic, as opposed to volitional movement. It's just a escape behavior, and they place a probe in the mouth right here. And what they do is they electrocute it, but they do so in a pretty specific way. They increase the voltage by 25% every time from 8 volts, and they move up in 25% increments. These are some mantis shrimp where they changed where they started administering the shock. So these are preliminary experiments where they're trying to figure out how much electricity was needed to elicit a response of 0.22 seconds. So how long it takes for them to exhibit a tail flick response within 0.22 seconds. So as you see, as they start to have the voltages, start to have the voltages start at higher intervals, they don't really have a diminished response. The curve is still there, almost irrespective of what voltage they start with. If they start with 8 in this case, or they start with 10, if they start with even at voltages at around 20, they still takes about 3 shocks for them to start reacting with sufficient speed to stop the experiment, which after they get to 0.22 seconds reaction speed, they terminate the experiment. They threw out animals that weren't exhibiting a response within, I believe it was 37 volts. If they didn't respond quicker than 0.22 seconds, they threw them out because they typically didn't respond at any of the voltages they tested as quick as 0.22 seconds. So what they found was that this tail flick response was susceptible to morphine administration. And this is actually one of the first claiming the paper, the first model of invertebrate morphine administration and pain conducted ever. I don't actually know if that's true, but the paper claims it. And what we're looking at here is that it's dose dependent. So as we step up the doses, the mantis shrimp take longer and longer to react to voltages. So where we have hundreds here, we're seeing a 100% increase in the pain response, in the response time. So we're clearly seeing a graded potential here, a U-shaped curve, a dose response curve to morphine administration. The more we administer, the more the response is delayed. And I believe it to be the case that over here we have low voltage shocks, and over here we have high voltage shocks. So at the very high morphine doses and at very high voltage shocks, we have 50% inhibition of 50% increase in response time to this shock. So 50% of, they probably expect 0.22 at this step. So it's quite substantial, at least 0.1 of a second increase in response time. And then over here, we're probably seeing, total, maybe they're not even responding to these voltages. We have our mean and standard error of how long it took for them to respond quicker than or equal to 0.22 seconds. More animals, each row represents an individual animal. And we see with naloxone is a morphine agonist, remember? So the opioid receptor that would dampen neuronal activity is going to be blocked by that naloxone. And the naloxone is not going to activate the receptor. We do have some amount of binding evinced by the fact that at the low intensity shocks, first 3 steps of the low intensity shocks on the right, we have a disinhibition of response. It's an increased response time. So one other empirical paper I want to talk about is a paper done by Zabala, and it's done by the same institute, same lab a couple of years later, but they're using a mantis for this. And there's a couple criterias that the mantis has a posturing reaction that it exhibits. And it has a criteria here. So the antennae are going to be pinned back, the mouth is going to be wide open, the prothorax, which is, I believe, above the thorax, is going to be raised. And so it's going to stick up its chest, raise its forelegs in the air, laterally flex them, pronate them, and it's going to stick out its wings, and then it's going to shake back and forth. And this is a way to prevent predation. It responds, it uses this response in a way to avoid painful action by a predator, we'll say. So there's some kind of mechanism here, whether or not it's a cognitive mechanism of recognizing a potential threat visually. They usually recognize these threats visually, but in the case of this experiment, they're administering shock to them, and they're exhibiting this response. So when you amp up the voltage, they only used about 16 volts in this experiment, so it's pretty low voltage. They noticed that the mantis, typically in the environment, you see from behavior A to behavior G, a graded response. The larger the threat, the greater amount of behavior you see in order from A to G that you get from the mantis. Now, if the threat is slowly removed, you're going to slowly have G go away. The swaying is going to stop. The mantis is going to keep all the other behavior the same. And then as the threat gets more remote, F is going to go away, and then so on until you have no posturing. So they have the mantis hooked up here to an apparatus, and they determined how much of a reaction they determined how much voltage it took to get a particular reaction, and they used no more than 16 volts. So they also administered morphine in one particular group, and they again did graded response where they had varying amounts of morphine. And here we can see the results. So 1 hour after morphine administration, we're having a full reduction of diagmatic response, or the posturing behavior. So morphine has an intimate role in regulating what this animal is perceiving as not only painful, but as dangerous to them. Now I want to talk about a book called Feeling the Pain and Being in Pain by Nicola Grahek. And this is interesting because in this book we see in a case of pain without painfulness, which is pain asymbolic. And pain asymbolic, in those patients, they can feel the thing that is giving them pain, but they can't actually care about it. So you have this condition where patients will come in, usually caused by lobotomies or cingulotomies. They'll come in and the experimenter will burn them. They'll prick them, but they won't be able to care about the pain. They won't change their affect. They won't change their behavior. They won't change anything about it. They'll even thank the experimenter after it's done. They doff their hat and walk out the door and have a good rest of their day, as if they weren't just being tortured. A deficit that these people have is that they are unable to care about menacing, threatening, potential threatening stimuli. So they have no affect concerning things that could harm them. So you can take a pin and prod it at their eye, not actually touching them, and they won't be able to flinch. They won't be able to react in any way. They won't get any kind of feeling that they're in trouble. So we take this back to the Mantis paper. We can see that even in vertebrates, this possibly seems to be conserved in terms of pain dissociation. The sensory information seems to be dissociable from the affect. And the affect can't be excluded from our definition of pain because, well, just common sense that if someone can't care about pain, they're simply not in pain. Here are some things that can cause pain on symbole besides lobotomy and cingulotomy. It's not to be confused with some pain disorders where you can't actually feel high intensity stimuli. You really can feel it. You just can't care about it. They say things like, I can feel the pain, but I don't care about it. Some people have questioned how good of a question this is to assess pain. While they can feel the pain, they can't really care about it. So many people have raised the argument that this is not actually pain, as I just brought up. Daniel Dent has talked about how lobotomies are so similar to morphine administration in humans. And we just saw that in praying mantises, we have decrease in reaction. And even in the shrimp, we see that automatic electrical responses, which are what tail flicks are able to be affected by these opioids. Whether or not they're binding to opioid receptors is up for debate. In invertebrates and mantis shrimp, probably not. In mantis, they probably don't have opiate receptors. So there has to be some receptor it's binding to get this effect. What it is, we don't really know, but morphine broadly seems to have behavioral consequences in invertebrates. Potential mechanism to connect these two things together, the affect of feeling like you're in trouble or being concerned about something, and similarly, the affect associated with pain, they seem to be related constructs and likely occur in the same part of the brain, because we see that they both are diminished when particular regions of the brain are damaged, removed, lobotomized, cingulotomized. So we see that morphine, lobotomies, they have similar actions in that the secondary order neurons that are responsible for the affect, they're not getting the necessary information to make that, to integrate that sensory information and make it relevant for the experiencer. And these are the multisensory neurons. We know that pain neurons are kind of tied into visual system neurons in humans. So there's some kind of pain affect associated with visual perception in all animals, honestly, that have eyes, that is. So we're going to turn to the question of whether or not insects particularly feel pain. So since Descartes, we've had a problem where we didn't want to poke around in the brain, especially in humans, because we thought we'd be, playing God or poking around where we shouldn't be poking around. But since Descartes, we've kind of swept those, those assumptions aside until recently, which it's kind of ironic that the reason we started doing neuroscience is because we didn't think the body had anything to do with the soul, and we didn't think animals had souls, but it comes to find out that the structures, especially in primates, analogous to humans, the structures are so similar we can't actually say that these animals are not experiencing pain. These mammals aren't experiencing pain, but it's ironic that we discovered this because we believed that was not true. So there's some very difficult things to get over when talking about insect pain. Some very interesting observational data from Eisman is that insects don't really modulate their behavior when they're injured. When they have a leg crushed, they continue walking with undiminished force. They continue doing routine behavior as they're getting eaten alive. They do all kinds of things that just seem like it wouldn't be the case that they would be doing that if they were actually capable of feeling pain, because that would be the only thing that they could be caring about. Now, this isn't definitive, because even though we have evidence for the goalposts are much farther off than that, obviously proving that someone is in pain, proving that anyone is in pain is nearly impossible experimentally. But proving that insects experience pain, it's not sufficient to say that since some insects can't modulate their behavior to diminish pain, since an insect can't lighten the amount of pressure it uses when it's walking on a damaged limb, just because evolution hasn't, selected that doesn't mean that they're not in pain, but it's pretty decent evidence for a lack of pain in insects. And we need to distinguish between nociception and pain. Pain is the subjective experience of being concerned about pain, about feeling a certain threatening or potentially threatening stimulus and caring a whole lot about it in a negatively affective way. Now, bacteria can respond to noxious stimulus with nociception. They can, detect chemical agents and they can move away from it. But this is not pain. And I mean, fruit flies have neurons. They have nociceptive neurons that do transduce, dangerous stimulus and elicit behavioral responses. But it's not clear that that is actual pain. And we need to not, put our, human intuitions about pain onto insects when we're talking about this. But broadly, we can consider pain, as an unpleasant sensory and emotional experience. You can't dissociate, as we've talked about it, the two. They're intimately entwined. If you don't have sensation and you're in pain, there are conditions where you can feel like you're in pain or have a sensation of wrongness without actually knowing what the sensory information is. That could very well be a form of pain. And I mean, it's the important bit, which is caring about the pain, feeling bad that you're in pain, but not being able to put your finger on what it is that's causing that. Whereas the reverse, being able to sense pain without actually caring about it, isn't really pain. Now, this caring about pain is what we want to identify in insects, not the ability to know that something is wrong, but the ability to care about that information. Most of this hinges on consciousness, whether or not insects are actually conscious, and most models that investigate mammalian consciousness are very inept for insect consciousness. Most involve, placing sticky tape on mammals and having them look at a mirror. And if they can look in the mirror and know that they're looking at themselves, they'll take the tape off without thinking about it. Insects, it's much harder to craft any kind of paradigm where this is the case, from divergences in many biological systems. Gregory Neely has done a paper where he shows that cutting off a leg of a fruit fly does decrease the pain threshold. How much pain does it take for them to elicit a response? This is a form of chronic pain. While they don't, limp or exhibit any characteristically human behaviors that are associated with pain, they can change the threshold, how much pain they're willing to endure, or nociception, to, you're not seeing them limp, you are seeing them prefer other sides of arenas or prefer something when normally if they weren't injured, they wouldn't have that kind of preference. So we can see modulation of behavior based upon damage, but there's no real way to cut through this and say that they're experiencing pain, although this is pretty convincing. This is pretty substantial evidence for some kind of phenomenon similar to pain in insects. Since we don't really have any positive information regarding whether or not insects have pain states, Wigglesworth has wagered that in kind of a Pascal type wager that we don't have the information. The risk is quite large in terms of potential suffering we could be putting in the world if we're using insects for pain research, and unethical paradigms where intense, death, suffering could be occurring. We should treat insects with the same deference that we treat mammals or other lower creatures that we categorize as such. An attempt to, reduce suffering in the world and reduce the amount of animals we subject to our experiments. It's really all I had for today. I hope you learned something. Thanks for tuning in.